Hi, welcome. I'm really glad to be here today to talk about being in the field with citizen science. And the focus of today's webinar is sampling and identifying two things that are kind of challenging to do in a virtual webinar, but I think that the team has come together and put together a pretty good facsimile of the real life experience. So without further ado, welcome. Cassie, we can move to the next slide. So you have seen this slide probably before. This is when we expect to see the emergence of mosquitoes in different parts of the country. And I would like you to shout out in the chat if you have mosquitoes where you are right now and if you've been seeing them. So just go ahead right now and identify, where you live and if you have found any mosquitoes. And we'll check that against this chart here. I know for most of the country, we're still right before mosquito season. I know that some of my friends in southern Florida have had them for quite a long time already. And yes, in Milford, Massachusetts, that's really interesting. Bob, when did you first see them? I'd be really interested to hear that. This is perfect. So some people are seeing them and some not, but if you look here please take a look at where you live in reference to this map, and you'll see that this is about when you should start finding mosquitoes. And what we would really like is a week or two before you're expected to see mosquitoes to go out and start surveilling so we can actually get that first appearance. Because Bob just said he just saw them for the first time on Saturday, late in the day. That's pretty cool. And Elizabeth said that she's starting to see them too. So anyway, and Ethan's talking about that they have them year-round in Hawaii, and I know that because I just got back from Hawaii talking to some of my colleagues there. So we'll be talking about that a little bit later today. Great, next slide, Cassie. So this is what we're going to talk about today. We're going to talk a little bit about the campaign and what our impact is. It's only been going on for a couple months. We don't have a big impact yet, but we're expecting to light up that map a little bit later this month. What we're going to talk about today is how to identify mosquito sites in the field. And I'm going to give a very short example of a citizen science project that's taking place now in Hawaii. And then finally, we have a guest scientist today who is going to lead us through how to identify mosquitoes. And for me, this is the super fun part of this webinar. A quick shout out to Globe Observer, because on the Globe Observer platform, you've been seeing this now for the last 2 or 3 months with clouds, mosquito habitat mapper, and land cover. And Cassie, why don't you introduce the new tool?
We have globe trees, and I know that Dorian's made a couple of observations already, but basically finding any tree that's about over 5 feet tall, I believe, and you go out and it walks you through making your observations and doing a tree height study for us.
Excellent. So this is the launch for this new app is just this week. And so we'll actually talk a little bit more in not next webinar, but the one after. We're going to be talking about how the mosquito habitat mapper data can be connected to the land cover and tree data. So stay tuned for that. So we'll go over that in a little bit more detail in about 2 months. Okay, next slide, please, Cassie. So what I wanted to do is I wanted to show you some of the data that we have collected so far as part of this campaign. And forgive me for this simulation because we wanted to save all our time for Krishna today. But next month, we will actually be, Cassie is going to lead us through how to access the data using the Globe website, how to visualize the data, how to make a video of your data, and how to then do some analysis using and export your data. So we'll talk a lot more about that next time. But this is the interface at globe.gov. You go to Globe Data and you select Visualization. Next slide. And this is the data that we have as of today. And you'll see that There are 7,704 sites on the map. We have more than 10,000 data points that are observable. So we're really moving forward and working hard. You can see out there in Hawaii a couple of the observations that I made when I was there last time. Maybe some of those are yours, Ethan, too. I don't know. But you can see that there are a lot of different places around the world where we're getting some data. And we're going to be talking with Dorian Janney, who has been leading the K-12 webinar series for teachers. And she has been in contact with a lot of these really fantastic teachers and schools that are part of this Globe Mission Mosquito campaign. So this is just a holistic view here of where we are, and think about how you can put lots of points on this map as well. Okay, Cassie, next one. Okay, I'd like to introduce Dorian Janney. Dorian is the campaign coordinator for K-12 for Globe Mission Mosquito, and she's just going to really quickly just give you a view of what she's been doing in the international sphere. Take it away, Dorian.
Hello there. Thank you so very much. So yeah, we just wanted to give you a little tiny glimpse of some of the flavor of what's been happening internationally with the Globe Mosquito Habitat Mapper. Every week, I take a look at the top submissions of data that have occurred over the past 30 days. And these are data submissions that are coming from Globe schools, Globe teachers, and what are kind of known as Globe virtual schools. So you won't necessarily see the citizen scientist data up here. But in looking at the more formal education programs, what I hope you'll see as you look at last month's honor roll, which we included in our newsletter, is that these data are coming to us from all over the world. And when somebody, a school makes it on the honor roll, it means that they have submitted multiple data sets. You might also notice that, well, I guess it doesn't actually say, but we have one site there that is from Medford Memorial Middle School, which is actually in New Jersey, and they don't have active mosquito season. One of the things we really like to impress upon people is that please use your mosquito habitat mapper even before your mosquitoes are active, because that is going to be very, very helpful in having you be ready and have identified some of these potential mosquito breeding habitats, as well as have you be ready to start identifying when mosquitoes become active in your location. And if I could have the next slide, please. I just wanted to show you a couple of pictures of these Globe students and their teachers in the field. On the left, where there is active mosquito season most of the year, we have from Princess Chulabhorn Science High School in Trang, Thailand. We have some students that are actively involved in finding larvae, and then they also did use the cell phone magnifiers to identify the types of larvae they found. And then on the right, we have some of those students in New Jersey getting prepared for active mosquito season by making their mosquito traps. And next slide, please. And I wanted to finish off showing you that one of our top countries who has been submitting Globe data through their virtual Globe schools is in Senegal, Africa. And here we see some of the teachers and students out there looking for potential mosquito breeding habitats. And indeed, they did find some of these habitats, and they also found some mosquito larvae. And there you can see, using their cell phone magnifiers, a picture of the siphon of one of the larvae. So thank you so much for letting me brag a little bit about these awesome Globe teachers and students around the world.
You have really great bragging rights, Dorian. Thank you. And such a nice picture there of a siphon. We'll be talking about siphons in just about 15 minutes, so I'm glad you gave us that segue. One of the things that we're interested in, and this may or may not pertain to you and your personal situation, but NASA scientists are interested in partnering with people who are actively collecting data. And one of the things we want to get is we need to get data spatially, and we need to get data temporally. And so some of these schools, like the one here in Senegal, they have enough data that they have collected from their neighborhoods in their town that there might be a possibility of beginning to look at that data in the context of satellite data, which is telling us about environmental conditions. So if you would like to do a research project and connect with NASA scientists, and if you're willing to take a couple measurements a week, please let us know, because we would like to support you in that endeavor. We're going to be contacting these folks in Senegal and connecting them with a research scientist from NASA in the coming weeks. So please let us know if you're interested. Okay, thank you, Dorian. That was wonderful. Next slide, please. So we have already done this because people have been writing in where they found mosquitoes. So I'm going to segue from now into the rest of the talk about where I have found mosquitoes. And you may remember that the last webinar, I was doing some workshops in Hawaii with some folks at a Globe school. I was working with some folks at the Osher Lifelong Learning Center, and I was also meeting with some scientists that we're collaborating with. And so I just for fun, I just wanted to show you some of the variety of sites that we have found mosquitoes in. One of the things that we found was the ubiquitous flower pots. The flower pots that have a saucer under them, like this little red one here on the right, was filled with Aedes mosquitoes. Okay, next one, please. And also, one of the reasons why the citizen science effort is so very important from a health point of view is that these little mosquitoes are finding what we call cryptic habitats in order to breed. And what that means is they're seeking out places that are not places that we would normally look. And here's an example of two people that are looking in a storm drain on a patio that was filled with Aedes mosquitoes. So I just wanted to show you the kinds of variety of places that we're seeing. So these are the container type habitats, what we call container habitats. All right, the next one is we also have lots of natural habitats. And if you remember in one of our workshops, we talked about the Aedes mosquitoes as being mosquitoes that are originally, their grandparents, their great-grandparents and ancestors used to live in tree holes before they became acclimatized to humans and well adapted to living close to people in urban environments. And so one place that I have found in Brazil and in Peru and also here in Hawaii that is a really popular place for mosquito moms is bamboo. So here's a stand of bamboo just on the University of Hawaii campus on the left. And some of that bamboo has been cut off, as you can see. And that little hole, it's only about 3 centimeters across, 4 centimeters across. And that is a perfect little habitat for these mosquito moms to breed their young. There's no fishes in there, there's no predators, and it's dark and it's safe. So you have to really think about all these different places where they like to raise their young. Okay. And then another place we found mosquitoes, mosquito larvae in Hawaii, were in these flowers. Ethan, I don't know if you know what flowers these are, because I don't. But inside these flowers have a little vase, a little reservoir of water inside their fluorescence there. And in there we found two larvae. So you have to really, once you start collecting mosquitoes and looking for them, you'll never see the landscape the same again, I promise you. Okay, yes, bromeliads. A lot of bromeliads are really fantastic mosquito habitats. And when I was in Brazil, we found a lot of epiphytes that were filled with mosquitoes. So very, very interesting place to look, and not necessarily something you'd first think about. Thank you, Ethan. Okay, next one, please. So here is one of our collaborating scientists. This is Dr. Floyd Reed. He is a scientist at the University of Hawaii, Manoa, and he is working on a bird biodiversity project, which we're going to be talking about just in a couple minutes. And one of the things that he needs, because he's doing some genetic research, is he needs live specimens. So when I told him that we were going around with working with mosquito habitat mappers, and one of the things we do is we eliminate the sites, he said, great, but please don't eliminate this site, because it is the perfect place for me to capture specimens for my research. And so you can see, if you wonder what a tree hole is, I never knew what a tree hole was till I started working, with mosquitoes. But here's a tree hole. It's just a little pocket in the tree. This is actually in part of the root of the tree, and it's a place where water collects. And here he is doing some sampling. In the next slide, you'll see what he found. And Cassie, could you run this video? It's just what I asked him, he said, I can look at mosquitoes. I don't even need to use a microscope. I can just look at how they're swimming and tell what they are. And so he was just showing me how there is differences between the Aedes mosquitoes and the Culex mosquitoes and how they swim. But he says, I can't really explain it. It's kind of a gestalt thing. I just kind of know after looking at so many mosquitoes. So, that is one thing too that happens as you begin to use the mosquito habitat mapper. Once you've seen lots of mosquitoes, you may not need to go through the key all the time because you're going to have an instinctive and correct feel for what you see. All right, great. Let's go to the next slide. And I thought this would be a little bit of a treat because we can't go out in the field and actually go and see sites. But this is a lovely video that talks about how they have developed a research design to sample and control mosquitoes that are which are largely responsible for some of the problems that they have with these endangered bird species. One of the things that's happening is that because there is climate change, the mosquitoes are moving up in altitude. And at this time, these endangered birds are living at high altitude, and they've been able to survive because there are no mosquitoes there. And these mosquitoes are carrying an avian malaria, so a bird malaria that is actually decimating the population. And so they're trying to see, since they cannot stop climate change, they're trying to see if they can actually do something with the mosquitoes so that these birds can survive. And this is some of the research that our partners are doing in Hawaii. So Cassie, we're just going to see, I'd love you to see the whole video, but I'm just going to show you about 2 minutes just as a teaser. And it'll be a good way for us to move into an introduction to what Christian is going to be showing us in just a minute. Okay, go ahead.
The stream is broken up into 10-meter sections, and mosquito larvae habitat is thoroughly looked for in each section. Stream surveys are done routinely as a way to standardize looking for mosquito larvae pools and to make sure we check every possible area along the stream. Dippers are used to sample the water and look for larvae, and any aquatic insect found is identified and recorded. Mosquito larvae require stagnant or very slow-moving water to survive in, so we're mostly on the lookout for small pools next to the stream, like these ones. These small rock pools have larvae in them. However, these are Aedes, and if you recall, we're after Culex species. Here's how we tell the difference. The mosquito life cycle consists of adult, egg, larva, and pupa. Here in the larval stage, the Aedes and Culex look very similar, except for the siphon. The siphon is a tube that allows the larva to breathe air. It's like their snorkel, and the Culex siphon is much longer than the Aedes. Here they both are in a pipette. It's a bit difficult to film such small individuals, but you can just make out the difference in siphon lengths. So far, we've found plenty of Aedes pools, but no Culex pools. Nothing. 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 Nothing again. Nothing. And as the season goes on,
So I really wanted to show you that because, for instance, Anna was saying in the chat that she's had some traps up and she hasn't gotten any mosquitoes. And that is the nature of the beast. They are a little bit vicarious in where they actually decide to lay their eggs. And so once again, just to reiterate what Dorian said, the zero data is really important. Where we don't find mosquitoes, when we don't find mosquitoes, is equally important as when we do find mosquitoes. So it's data that is important and we love. So don't feel discouraged if that's what you're getting. Okay, there's just one other little section just to talk about why we're interested in this data in Hawaii that Cassie's going to show you now.
The akikikis are insectivores. Here, one enjoys a caterpillar. With less than 500 individuals, the Kauai Forest Bird Recovery Project monitors them by color banding adults and fledglings to determine survival and distribution rates. Having such low numbers, it's imperative to monitor them and try to reduce as many threats as possible, such as rats, non-native birds, and of course, mosquitoes. During a routine stream survey, Theo awkwardly suggests I check for mosquito pools up this thorn-infested drainage. I oblige. Ow! Jeez, oh Pete! Yep, Culex. Second and third.
Those are the instars we're looking for.
Yep. Ow! Ow! Okay. We have a Culex pool we can treat.
Great. So what did you notice about the places that they were looking for mosquitoes first, and in cases they looked at mosquitoes second? You know, in one place they were finding the Aedes mosquitoes, which were found in the little pools around the stream, but the Culex were found in a very different kind of habitat with a different kind of nutrient status. And so this is another reason why this kind of data is really important, because we're talking about what is the habitat like, where are they found, and who's there. And so I just thought this was a really very sweet video. And if any of you are involved in education or teaching, I actually thought it was a brilliant little piece that talks about how research designs are developed and how research is completed in the field. So take a look at the whole video if you're interested. All right, next slide, please. Okay, so it is my great pleasure to introduce our guest scientist. This is Krishna Vorhaide. She is at the School of Natural Resources at the University of Nebraska-Lincoln, and she is a bona fide mosquito expert, in my estimation. And she is going to use the magic of video science and a very cool little tool, which is a digital microscope that connects to her computer, in order to give you a firsthand look at how an expert identifies mosquitoes. So, Krishna, when you're set up, Cassie will transfer the
okay, to view my screen.
Yes, thank you.
Okay, hang on just a second. Gotta figure out what this is. Okay, can you see that?
We see a red screen.
Yeah, okay, I haven't gotten that. So the first thing I'll do, because it's pretty important, is to go over what a mosquito larva doesn't look like, because there are a lot of imposters. So this is a midge larva, and you can see that it's got a head and a abdomen. Here, it's gonna be hard for me to point it. Can you guys see my cursor here? And then it's got a tail segment, but it doesn't have a siphon. And here's another one. You can tell it's more worm, much more worm-like. It's still got some CD on the end, but there's no siphon structures, and the abdomen segments aren't very defined. So here we'll move over to a mosquito larva. And you can see, let me get up to the head part here. That's it's kind of decimated from being stored for so long. This is a couple of years old. I found it just like those guys found it in the video, running around, dipping a bunch of times. Let's get down here. This is the important part right here. You can see, here's the siphon, and here's the anal segment. This little patch right here is called the saddle. That's sometimes used to identify to species. In this particular specimen, we can tell it's an Aedes because the siphon is short and pretty stubby, but we can't see another one of the identifying features, which is the pecten teeth, which I'll get to in the next specimen here. So this is another head, mosquito larva head. And right you can see the pecten teeth right here on the underside of the siphon. And that's often how we determine species of Aedes. This is another Aedes mosquito larva. And I think this is Aedes japonicus, which is invasive to my area. And I think that because you can see there are these pecten teeth here that are all in a row, and then there's a gap, and then there's another one right here. And that's pretty typical for that species. I'm also trying to keep these things from drying out. They're in this tray here. I'll show you one more and try and get a better look at the, oh, here's some better pecten teeth. And these are really hard to see if the mosquito larva is in the wrong position, so you kind of have to twist them around with your tweezers a little bit. Can you guys see the pecten teeth? Oops. What is going on? So here's a row of pecten teeth on one side of the siphon, and here's another row of pecten teeth. And then there's a little hair right here you can kind of discern. And another distinguishing feature we use is this comb scales, which should be right here, which are hard to distinguish in some of these specimens because they're old. They've been sitting around in alcohol for quite a while. Now I'll give you an image of a typical, you just dump your larva out. And so there's a good head capsule. There's a perfect thorax with all the hair. CD, we call them. There's the abdominal segments. If you want to count them, there should be 8. And then there's one on the end that's got the saddle. And then here's the siphon, and it's sticking up towards you, so you can't see the pecten teeth. So you'll have to flip it over and see if you can. But if you only want to go to genus, we know this is an Aedes because it's a pretty short siphon. I don't have any culex to show you all the specimens that I gathered in June. One of these specimens is from June of 2016, and one is from last September, and they're all Aedes. They're pretty voracious little things. Did you want me to cover anything else? Or does anybody have any questions?
Yeah, I think this would be a good place for us to open up the chat for questions for Krishna. And you mentioned the saddle, but I really didn't see where it was on the specimen. So if you could just show us, maybe if you could go through maybe your best specimen and just show us the morphology and talk about the different parts, that would also be really useful. I've been typing in, what is setae, what is comb scales, and these kinds of things in the chat, so people have an idea of these scientific terms you're using. Yeah, but if you could show us, like, for instance, the saddle, that would be really great.
Okay, so can you see my cursor, did you say?
Yes.
This right here is the saddle. It's on the anal segment, which is this right here. On some mosquito larvae, it encompasses the entire segment. On some, it only goes halfway. This one only goes about halfway. So when you get down on the key to more specific features, that'll be one of the questions. This right here is the siphon. And these are the abdominal segments. Let me move. It's a lot easier to use this microscope if I just hold it. Okay, and then there's the head capsule. The mosquito larvae always have a head capsule that's wider than the thorax. if you find something that doesn't have a head capsule that's larger than the thorax, it's something else. And then there's this, this isn't a good example of a thorax, but there's always a broad one. And then there are little patches of setae, which are the hairs coming off of each. If you look at these things up close while they're swimming, they're sticking off to the side. They're not all discombobulated like this.
Mm-hmm. I think it's really interesting if you look at what's being called a saddle here. If you sort of turn your head upside down, it looks like a saddle on a horse, and that's obviously why they call it that.
Mm-hmm.
And one of the diagnostic features that you will see, I should say, in the app, there is a key for determining what kind of what taxa of mosquito that you find. And one of the questions they ask is, does the saddle go all the way around the abdomen, or does it go halfway around like you see here, or is it pierced by hairs? And you can see in this example that the saddle just looks like a saddle, and it doesn't go all the way around. So that is another one of the features that you get to look at and determine what you have.
So here's a head capsule and the thorax again with the hairs. And then right here, these are the little antennal structures. There's one there and one here. And I believe this is a little eye bud. And there's one over there also. Another diagnostic feature that happens pretty far down in the key is the head hairs. We won't get into that. Those are pretty hard to determine. And then you can see the abdominal segments. They're pretty distinguished. If you look at a midge, they're like a little flat worm, and these ones have definitive segments. And then there's the siphon again, and you can see the anal segment behind there. Let me try and flip this one over and see if I can make it look any different. Well, I kind of messed them up.
Mm-hmm.
That's going to happen. Yeah, it looks a little different. You can see the anal segment a little better. You still can't see the pecten teeth. I'd have to really manipulate that guy to be able to see the teeth.
Mm-hmm. So here's a question for you, Krishna. Mm-hmm. And of course, you're an expert, and I know you have looked at thousands and thousands of mosquitoes because I've seen the ones you've sent us for the AI. If you have a specimen and you can't see the features that you need to use to determine what species you have, what do you do?
If you can't see them definitively, then you consider it an unknown.
Right.
Maybe you can get it to genus. Mm-hmm. Which isn't that difficult, but if you can't identify it to species, then you just can't.
Right. And it's important. I wanted to point this out, Krishna, because you're an expert, and yet this would be a specimen that you might not be able to take to the species because the comb scales are not clear and the other features are not clear. And so you're going to find specimens from time to time that don't have the features that you're looking for. You might be looking at an immature specimen. Mm-hmm. In this case, you might be looking at a specimen that's been degraded because it's been sitting in alcohol. And it's Krishna's fault. She lives in Minnesota, so she doesn't have any fresh mosquitoes right now. But this is not a problem. And in the key, it's still really important if it's Culex or if it's one of the species of Aedes. And so you can actually, in the key, you can say, I looked at it, I can tell from the siphon shape and some of the other features that it's definitely an Aedes, but I can't tell if it's an Aedes albopictus, which is the Asian tiger mosquito, or it's Aedes aegypti, which is the yellow fever mosquito. Both of those are responsible for all kinds of diseases around the world. Mm-hmm. But just knowing it's Aedes is important. And so in the key, we know that, and you can say, I looked at it, all I can say is Aedes. I can't make that distinction. And that's also really important data. And it doesn't mean that you're doing it wrong or that you don't have the skill. It means you have a specimen that can't be identified. Mm-hmm. So thank you so much for bringing in a specimen that just to let people know that that's really okay. Mm-hmm. And also, talk a little bit about looking at fresh specimens versus specimens that are preserved, because I think that's also really important, because this looks a little bit more scary than it is.
Yeah, well, the fresh specimens are still fresh, so they've got all their features. It's really hard to identify them when they're moving, so you have to get them to sit still. You either have to, sometimes I just cool them down in the refrigerator first. Sometimes you just put a drop of alcohol on them, but you'll be able to see the hairs will be more lively, and they won't be, the body, it just won't be as degraded, so it'll be a lot easier to see. One thing that I noticed, right here are where the comb scales are. Can you guys see that little row of?
Yep.
And that when the specimens are fresh, those are a lot more easy. They're a lot easier to distinguish. And there's different.
Great.
Yeah, there's different arrangements for the comb scales. Sorry, I keep fuzzing it out here. Okay.
Well, you're holding it by hand. So yeah, so basically with the comb scales, and you can see some really great, when you go to your app, if you have not yet tried to identify mosquitoes using the app, there are lots of pictures and diagrams that will take you through and say, do you see this or do you see this?
Mm-hmm.
Do you see this or do you see this? And they will show you pictures of what the comb scales look like. And, if they look like, whether or not they look like thorns or whether they look like pitchforks, and if they are in a patch of a whole bunch of messy scales in all different directions. I saw some of those in the Amazon, and they just look really weird.
Mm-hmm.
Or if they're in nice tight rows, like we have with the 80 specimens that we have here in the States.
Mm-hmm.
So it's not as daunting as it looks. And I'm monitoring the chat, and we don't have any questions. And since we're taking up Krishna's valuable time, please ask her some questions so that she feels like you have that she can help you out, because she's got a big heart.
Well, one of the things I noticed too after identifying a bunch of these is that the 80s mosquitoes tend to have one or two, aside from the pecten teeth arrangement, the 80s have one or two hairs generally coming from this area of the siphon. If it's a culex, there are usually multiple hairs, possibly up and down, and they look pretty random. I wish I had one, because they're pretty easy to tell apart. And if it's a culicetta, which also has a really long siphon, they have hairs right here at the base of the siphon, and no other genus has that yet. We'll see.
That's right, because there are 3,500 plus species
Right.
of mosquitoes, and so when you say yet, she's speaking as a taxonomist that understands those things. There are some great questions here. I'm going to read them to you so you can just keep focused. One person asked us, how long is this specimen here?
Oh, that's a good question.
And an estimate is fine. You don't have to take out your
Well, I'm bad with measurements. A couple of, I would say maybe 3 or 4 millimeters.
Mm-hmm. So basically like half a centimeter, a little bit less.
Yeah, right.
Yeah. They're really small, but they
Less
are
than a quarter inch.
Yeah, but they're small, but they're macroscopic.
Mm-hmm.
And so, you are able to, you can actually look at, once you have a refined eye, you actually don't even need to use the microscope to make a preliminary breakdown of whether or not it's probably a culex or probably an Aedes.
Mm-hmm.
To make the final determinations, you do need to use it, of course. But another question is there a suggested magnification? And this is a really good question. We have been using 60 power microscope, clip-on microscopes in our educational programming. That magnification is good to get you to at least the genus level of Aedes. I have found some difficulties with the 35 to 60 power seeing comb scales. Have you been able to see comb scales at that level, Krishna?
Mm-mm.
Yeah.
Or pecten teeth, really. I mean, you can discern them if you know that they're there, but if but they're hard to see otherwise.
Right, you see it more as a dark line, the pectin.
Mhm.
So the suggested magnification is at least 100 power. What is the power on this digital microscope that you're using here? This is 1,000 power?
1,000, yeah.
Yeah. And the clip-on microscopes that we've been using in this project are around $8 or $9. And just recently, we have been able to find these digital microscopes that you can clip to your computer or to your pad or mobile phone, and those are running between $19 and about $30. And so that with that kind of magnification, you'll be able to see all these features in great clarity. So there's no guessing, but not everybody can have that kind of magnification. So that's why we're saying we're really happy if you just say there's larva, and then we're happy if you say it's some kind of Aedes or it's a Culex. But if you want to make those finer distinctions, and I know, for instance, in some of the research that we're doing as citizen scientists working with scientists in Oklahoma, they really need to know for their research project if the mosquitoes are Aedes albopictus or Aedes aegypti. Both of those are mosquitoes that are invasive mosquitoes that are now moving northward in response to changes in climate. So for the citizen scientists that are working there on that project, we are actually asking people to see if they can get access to a higher magnification scope. Let's see. Okay, so we asked about that. Okay. Are there any tricks other than repositioning the larvae and using fresh larvae for finding the comb scales?
Those are the only two I know about. If you come up with one, let me know.
But you have to know where to look, right?
Yep, you have to look on the siphon, on the underside of the siphon. Just repositioning them.
Mm-hmm.
I think if they're fresh and you have them in a bigger puddle of alcohol or water, sometimes they're easier to move around with your tweezers.
Mm-hmm.
I could try that now, but you won't be able to see it because the only thing I have is this microscope.
But go ahead. You can go ahead and show us how you turn it over. That would be very good, I think.
Well, you won't be able to see it. Oh, wait. Let me see if I can do this. Oh, fun with a microscope. Okay, here we go.
Yeah.
So then you take your little tweezers. Wait, where did he go? This is hard.
It is.
I mean, trying to do it.
There you go. I also have to say that poor Krishna only got this microscope on Monday, so it's the first time she's used it. So we really appreciate her, doing a demonstration on a brand new instrument.
Yeah.
So, she's using tweezers. We also, when we've been working, we found that just using a toothpick works really well just to flip them over. You don't really need to have tweezers. And sometimes a toothpick, because it's wood, might also keep it from damaging the mosquito a little bit. I also thought your comment, Krishna, about the amount of liquid to suspend your specimen in is really important. And I have used several different kinds of clip-on microscopes. And I've also used the same digital microscope that you're using. And what I found is that, you can't say for sure, oh, this much works. It's really going to depend on the focal length that you have, right, and the resolution of your microscope. And so you can just experiment a little bit. And in the instructions that we have in the app, it says you can just take a little piece of Kleenex or paper towel. If you want to be scientific, you would say use a Kim wipe. And, you just take a little bit of water away from the sample until you get it so that it's suspended in the right amount of liquid so that you get, a good refractive index and a really good specimen. Gary asked about the name of the microscope. And I want to tell you that I tried to answer that question, but if you just look for a digital USB microscope 1000 power, you'll see a whole list of them if you go to an online store. And if you look at the pictures, they range from $60 to about $19, and they're almost all the same. Some of them have Bluetooth, some of them have a connector that you can use with a phone, but they're all basically the same, and they don't really have a name. This one here that she's using, because I bought myself one too, it just says digital microscope, and there is no branding. It's just digital microscope made in China. So hopefully that's useful, but I have had no trouble finding it. You're welcome, Gary. Okay, we have time for one more last question for our visiting scientist today. If anybody has any other questions. And while you all are thinking, I have one more question, Krishna, about you were talking a little bit about the imposters, some of the mosquitoes some of the specimens that are aquatic invertebrates that people find, and not very often, but once in a while, people will take a picture of that and upload it and say that they think it's a larva.
Mm-hmm.
Could you talk just a little bit more? You mentioned briefly about the head of the microscope, the head of the larva. Could you just talk a little bit more? Once again, you did it early, but I think that it's good to refresh what you said about those imposters again, because it really is tough. When you've never looked at these before, how do you know what you have? And Krishna can give you some really good tips on that as well.
Yeah, so this, you can see that the abdominal segments look completely different. This one is emaciated, but still, if it's a midge, it's going to not have the defined abdominal segments. There are no hairs on the abdominal segments here either. There's no thorax. There's no broad thorax here. There's a sort of a there's sort of a head capsule. And then we have this thing at the end that's got some setae or hairs. And there's no siphon, and we can't be confused because Anopheles larvae also don't have a siphon. So a lot of times you'll look at a specimen and see that it doesn't have a siphon and assume it's Anopheles, but then you have to double check, does it have a thorax? Does it have setae? Can you see that the abdominal segments are distinguished? I'm not sure what kind of midge larva this is, but midges share a lot of habitats with mosquito, particularly chironomid, and also I can't remember the scientific name, but the little blood midges. So they really like a lot of the same habitats that culex like, because there's no oxygen in the water and it's got a really high nutritional content. So they're red, they create their own iron. Here's another midge, and you can see, it doesn't have the same abdominal segments as a mosquito larva. It has no thorax. That's one of the more important parts, is that it just doesn't have any shoulders. And then at the tail here, again, there's no siphon. They do look awful wormy to me. And they swim differently too. If you can see them in the habitat, you can see them squirreling around differently than a mosquito larva.
And that's exactly what Dr. Reed was saying too, is that he can look at them, and just by seeing them swim, he knows that they're mosquitoes right away. And then he actually can look at how they swim and figure out probably what the likely genus is. So once you look at a bunch of mosquitoes in habitats, it's very easy to identify how they swim. And when we were first developing this app, and this is back in 2015, and we wanted to develop something that kids could use, that non-scientists would be interested in doing. And we were talking to little kids that were living in Barbuda, which is a little island, a leeward island in the Caribbean. And the kids said, "Oh, you mean wigglers."
Yeah.
Because they have a very distinctive wiggler style. And all these 6- and 7-year-olds, 8-year-olds, 9-year-olds, they all knew that these were wigglers, but they didn't necessarily know that they were mosquitoes.
Mm-hmm.
And then once they develop a little bit further and they become a pupa, they have another distinctive way of locomotion, and they tumble, and they call those tumblers.
Mm-hmm.
So it's really quite fascinating. All right, this is a great picture of a head here.
Mm-hmm.
Okay.
And a thorax
thorax.
The mosquito larvae have these hairs coming off the thorax. If it doesn't have that, then it's probably not a mosquito.
Mm-hmm.
And you can also, just for fun, it's not diagnostic, but right here are their feeding brushes. Can you see where I'm circling?
Oh, yeah. Yeah.
So they have these little fans. Each one of these is a little fan, and they wriggle around, and they, depending on what type of feeder they are, they either scrape the edge of their container or they suck the little microscopic algae out of the water with their little mouth fans.
Great. Krishna, I want to thank you so much for taking time out of your busy day today to talk with this international audience of citizen scientists and share your knowledge. I hope you'll come back again and give us a refresher when we have some fresh specimens. Would you be willing to do that?
Yeah, for sure.
Awesome. Okay, Krishna, thank you so much. I just want to spend a couple minutes making a couple announcements. So Cassie, if you want to bring us back to the presentation stack. Okay. So the first thing I want to do is to let you know that as part of the NASA Citizen Science Globe Mission Mosquito team, you are cordially invited to attend a science lecture. It's going to be online. It's by one of our colleagues that's working in Oklahoma. He's going to be working with a group of citizen scientists in his work. But the title of the talk is Mosquito-Borne Disease Surveillance in Oklahoma. And the information will be in this slide. If you go to the Globe Mission Mosquito Campaign website and you go to stored webinars, you'll see all this information. And if you're interested in listening to a 1-hour talk talking about how mosquito data can be combined with data from NASA satellites to better understand when we might have outbreaks, I think it's going to be a very interesting talk. He's a very cool guy. Okay, the next announcement is the most exciting announcement. Go ahead, Cassie. And that is Citizen Science Day is coming up. It's April 13th. And the Globe Observer program has decided that they're going to support Mission Mosquito as the data challenge for everybody who works with Globe Observer. This gives us a chance to light up our map with lots and lots of observations. And we hope that you will participate in this data challenge. It is running from April 7th, which is a Sunday, till the April 13th, which is the Saturday. The last day is a Citizen Science Day. And we are asking you to make as many observations as you feel comfortable doing. And we will be providing award certificates for those participating in this data challenge. We're going to be having some certificates for making the most observations, the most complete observations, because you can just look at and say, yes, there's water, no, there's water, I tipped it out, right? Or you can go a little bit further and talk about the larvae and how many there are, and then even identifying them. So there are different levels of participation that you can be part of. And then also, I thought it'd be really good because we've got some fantastic photographers that are working with us. And we thought we might even give an honorable mention for some photography that's submitted. So I hope you will join this challenge. What we're thinking is, just like in clouds, we don't want more than maybe 10 observations, 10 different points a day. We don't want someone to go out and do 100 points one day and just do very, superficial work. But so don't try and do lots and lots in one day. But what we want to just really build up this data set so that it can be used by yourselves and by public health officials and by scientists. So please join our challenge. We'll be sending out a ping to let you know more about it. We're going to have a URL up probably after tomorrow, and Cassie and I will be sure to send that out. So I want to thank you so much for coming to our webinar today. I want to thank Dorian for her presentation. I'd like to thank Krishna Warhide, our visiting scientist today for her presentation. And we'd sure like to hear from you. Let us know what you thought. Next slide, please, Cassie. And this is the last thing. That here is my email, and I'd like you to scribble that down because I would really like to hear from you if you have questions. We have a little bit more of the Hawaii mosquito story is posted in the blog, and the blog is not set up so that you can actually interact with the blog. But if you send me an email, I will put it in the blog and we can develop some kinds of discussions because I think there's a lot of cool things we can talk about. Okay, the last thing I want to say, because we have one more minute, is that if you felt like that was too much information from Krishna, just know that all the things she talked about is both in your app, and also there are slide sets that actually will take you through the identification of mosquitoes, and we can make sure to make that available and as a link for you. So if you have any other questions, let us know. I think this is the end of today's webinar, and I want to thank you for your attention, and I certainly had a great time. Thank you.
Thanks for having me.
Thank you, Krishna.